Andre, thank you very much for this lovely and overdone introduction. I don't deserve that of compliments. I would like to say before I start that I'm really glad to be here at Češť, where not in this hall because we were at that time in a different castle, but I think I started visiting Češť in 2000, I believe, for the first time, and then 2001, 2002. And then several other times in between, and it was always a very lovely experience, and I'm very happy to be back at Češť, and I will also be at many future Češť as well. I'm very convinced about that. So it's a very good situation to be. Today I will be talking about our research after I came back from Norway to Vienna. We got some research project funded in Vienna, and I must say we were also very lucky with the constellation of domain partners who were, for some reason, at the same time in sync with us. So they sort of were ready to have the data we would need for the visualization purposes around the same time when we were starting developing our technologies. So this all starts basically with a short metaphor. And that is what we are doing is something like what is shown in this picture. Here Alice is drinking a drink me potion, which gives her some special skills. And those skills are to discover the world of small things for a certain while until she grows back again. And metaphorically speaking, this is what our technologies is about. We are also allowing you to see those little things which are way too small to see with eyes, too small for being seen by a microscope, even electron microscope, but then again a little bit too big for being seen with X-ray crystallography. So it's something called a mesoscale, which is up to now very hardly measurable, and therefore it is somehow procedurally modeled with biologically correct knowledge. So what we see here is a data set which has been created by our partners from biology, from the Scripps Research Institute in the US, where they have a very active research in HIV, and they call themselves Integrative Structural Biology Research Group because they are integrating all information which has been through reductionistic research acquired in several research labs, and they are putting this information together to form an entire structural model so that we have all present knowledge, at least about the structure, about the HIV. So what you see here in red are the proteins of the blood plasma where this HIV virus is embedded in, and this is how the HIV basically looks like. So before we go to the description how it is built, I would like to just give you a small appetizer where we would like to be heading in the near future. HIV is a complex virus, but still quite small. And on the top, you can see this indication of its size. It's by diameter 100 nanometers, and here we have a size of 15 billions of atoms which are comprised for this omnipresent E. coli bacteria, which we would like to model in the next together with our domain partners, and also to be able to visualize it effectively to convey its internal architecture. As you can see, it is much bigger, not really maybe in the size because it's 2 microns by the long axis. But in terms of volume, it is much bigger. I think it's 20 millions of atoms versus 15 billions of atoms in dataset size. This has not been rendered. This has been rendered, but manually by David Goodsell, who is a great visual storyteller. He's writing books about which is popularizing biology, and at the same time, he's a scientist and works at the Scripps Research Institute, which we are collaborating with. So we have in our team the illustrator at the same time, so that we can make some good design aesthetic choices when it comes to rendering. So our data set comes from the knowledge of biology, from measurements, but it's somehow derived, is the second order generation of data in a way, because it is procedurally modeled. So the knowledge which we have about the HIV in this case is coming from microscopy, from the higher scales, where we do not see crisp structures, but we see distributions, and we can measure quantities, how many of particular proteins or other macromolecules, for example, other molecules, are present in which area, in which zone of the virus. So we don't see them clearly, but we know that they are there in particular areas. We have a statistical distribution of it. Then at the same time, for all proteins, which are the machines which carry out all the function of life for the virus and for all living species, there we can see them most of the time, or in many times, with so-called X-ray crystallography, which is able to reconstruct the 3D extents of a protein, and at the same time, its chemical composition, meaning what type of atom is at particular location. So if we are looking at this, this would be all proteins which are then packed in these compartments. We know that the HIV has several levels of compartments. So one is outside, one is beyond a phospholipid membrane. So inside the phospholipid membrane, that's one compartment. And then, as viruses have, they implement some kind of a nucleus, but the nucleus is not composed of phospholipid membrane, so it's not lipids, but it's actually proteins which form this structure, and that's called the capsid of the protein, where the core of the virus is stored, the RNA in the case of the HIV. So all this information needs to be basically packed through procedural modeling process, and at the end we have a data set which is very complex for standard computer graphics systems such as Maya to be rendered interactively. So here you can see it consists, only the particle consists of 20 millions of atoms, and if every atom would be a tessellated sphere, it would be a lot of polygons to render interactively. So, but there are natural ways how to overcome this problem of rendering this type of data set effectively, but these techniques are not implemented in Maya, for example. So our first step was to get in a collaboration with these domain partners to take out what the computer graphics offers and make the rendering as fast as possible. And here is in principle shown what we are doing, and this is not this is a collection of research ideas which have been done by many other people and some other ideas coming from us. So we are basically rendering everything on the GPU, and we are also generating a lot of geometry on the GPU directly. So we are trying to minimize the amount of vertices coming to the GPU rendering pipeline. And basically, as you have seen these proteins previously, the proteins consist typically of, let's say, 5,000 atoms. And each atom would consist of a lot of vertices if you represent it by triangles. So, but instead of representing it by triangles, we can use so-called procedural impostor techniques, where we just render two triangles or one quad, or maybe one triangle, that's also possible. And then in fragment shader, we compute analytically, instead of intersection with a triangle at that location, an intersection of a sphere, if that sphere would be there and would somehow be geometrically related to the triangle in that particular location. So you can see here that if this would be the plane of the triangle or of the triangles, then we are evaluating basically, instead of the real intersection at the plane, we are evaluating what intersection are we having with the sphere, and then based on this information, we are changing the the shading at that particular location, and the depth, so that we have a correct results at the depth test. That is happening in the fragment shader, so quite late in the GPU rendering pipeline. Before that, we have other stages. So, for example, in the vertex shader, you can imagine that we are just sending there one vertex and an orientation per protein. So each protein consisting of 5,000 of atoms is represented just by position and orientation. And then we have on the GPU some kind of a shape library, which can then be used to, at a particular location in space, to instantiate this geometry which is stored on the GPU for each protein type separately. So that's the stage I'm talking about here, where we use the geometry and tessellation shaders for emitting geometry, these quads I was talking about, which are necessary for the fragment shader, so that we have it for every single atom available. Okay, so this is the first step of our rendering pipeline, which leads to significant increase of performance from minutes or tens of minutes to 60 frames per second suddenly. But that's not the only performance accelerations. We can deal also with the level of detail. And here, for two reasons, both for performance reasons and also for reasons of what do we want to promote in the visualization. So if we are, for example, too far away from a particular protein, we are no longer interested in seeing every atom of it, and instead we would like to see the overall shape. So therefore, the level of detail is not only computationally more efficient, but also it promotes a shape which is more dominating those qualities we want to see at that scale. So here we have one heuristic which we did, very simple one. Imagine that if we have the protein stored in such a way that from the distance of the center of mass of the protein, we sort all the atoms. We have somehow a heuristics that several atoms which are nearby in a linear array would be also nearby in a 3D structure. This is a heuristic which doesn't hold every time, but for kind of spherical-like shapes, it holds quite well. So once again, we sort the atoms based on the distance from its center of gravity of that protein. I mean, gravity not in the physical sense, but in the sense of geometry. And then once we have this ordering, then we can skip some atoms to be rendered at all. Every kth atom can be skipped, and we can increase the radius of the other atoms afterwards in order to preserve the shape. And by this we render much fewer spheres, but at the same time we gain computational power, and we also have a more understandable shape, less details which are not so interesting at particular distance from the viewer. Okay, so this was how we are accelerating rendering of proteins predominantly. There are other structures which are somehow complicating the life, but very important for the life at the same time, and those are the linear strands. Typically, this would be genetic acids like DNA or RNA. You can see it here on the manual or handcrafted rendering. All these yellow strands here, how to populate the atoms there. This, for example, this level of detail scheme, this heuristic will no longer work in that case, for example. So what we can do, we do not have the rendering pipeline doesn't enter only one point at the location per protein, as it was the case for proteins, but for the linear strands, we have a parametric curve which approximates the position of this nucleic acid. Whether it is single strand or double strand, it doesn't matter at this moment. And then we reparameterize it so that every cycle of or every revolution, we have points along this parametric curve. So this is an arc length reparametrization, so that we have control points at locations which we want, which are somehow related to the biology. Once we reparameterize that, then we compute for these points normals, so that the variation of normals along this curve is minimal. And once we have this, we, with the tessellation shader again, as we did previously, we populate within each revolution, we populate the base pairs which are creating this, or bases in case of RNA, which are creating this one revolution. And once we have that, we can again look up in the library of shapes either a base pair combination or just one nucleic acid base at that location, and render those atoms which correspond to that location. Okay, a great deal of performance can be also saved by the good occlusion management scheme, because these scenes are, on one hand, we always talk about biology that 50% of our bodies, or how many percent depends, consists of water. And maybe you have noticed, maybe not, but we are only showing the dry mass. Only the macromolecules is what we are showing. We are not showing any small molecules like ions, water molecules, and things like this. So if we would be rendering all the waters, the scene will be totally dense. If we leave them out, theoretically, we leave out 50% of the space, or maybe more, but still the scene is very dense, which means that if we are not dealing with transparency, we are very early on terminating our array at some protein, and that protein typically occludes a lot of other structures which are behind. So it pays off to have some occlusion management scheme which doesn't send into the rendering pipeline proteins which are guaranteedly not seen. For this, we can, for example, use the hierarchical Z-buffer technique, which is an old technique implemented in Compute Shader. Do you guys know what is a hierarchical Z-buffer? Hands up. Okay, so it will be explained very shortly because it's a cool technique, although it's very old. So here, the depth buffer is used for fragment depth test, right? Usually. But here we can use it, if we have a hierarchical depth buffer, we can use it for discarding entire objects. So imagine that we have a depth buffer and we want to test against this depth buffer whether some object is in front of the existing depth buffer or behind. And entire object, not fragments. So we can create a MIP level of depth buffers with a max function. So we are storing from the 4 pixels in the next level only the one which is the most distant one. And by that we create a approximation of the depth, but the overall size of each of the fragment or of each of this kind of pixel at this resolution represents approximately the size of the objects you want to test against. So here you can see that one protein would fall into 4 texels in this case, if it's a texture, and we can test within 4 texels whether this entire object is to be rendered or it's guaranteedly not visible. So this is it's a very nice technique, but the question is, how do we create this depth buffer at the first place? So usually you can use what has been rendered in the previous frame, and you are moving the camera just a little bit, so probably everything what has been rendered, or most of it, will be seen. So I can just take those molecules which I know that have been seen in the previous frame, and of course transform them, and then render those, and then create this depth buffer, and then test for visibility of all those other molecules which have not been seen in the previous frame. So this is possible to do, and it accelerates the performance by 50% in our case, roughly. We have also another level of occlusion management, and that is once we decide that, okay, we are going to render this molecule, within the molecule there are still these 5,000 of atoms, and maybe you need to render only 30 of them, and then the rest is already occluded. So for that we are using typical fragment discarding technique. However, we are dealing with these imposters, don't forget, so not with the real geometry. And therefore, we are having something which is called conservative depth test. So we have a depth which is the depth of the closest possible fragment of the atom of the sphere which we are rendering, instead of the real depth at that location. Okay, good. So by this, we can suddenly gain performance by that extent that from rendering frames for a long time, we gain interactive performance on regular commodity graphics cards. Here you can see a DNA, complete DNA of one of the simplest bacteria, which is also a model organism. It's called Mycoplasma. So this is bigger than a virus, than HIV. It has in diameter, I think, 400 nanometers, and it's an organism which is used in the project which is called Minimal Genome Organism, which is like a computer or human generated possible organism which would be able to survive. So in the genome, all the proteins are encoded which are necessary for the most elementary life functions. So this Mycoplasma is very close to that. So it's the smallest bacteria maybe, but anyways, it's a very large data set. How many have yesterday not seen the demo of Tobias, the HTC Vive demo? Who hasn't seen it? A few. So I will run this very quickly. This is not a immersive setup. It's a desktop system, but you can here see how this can be applied. So you can have basically a kind of a learning tool where you learn what does blood plasma consist of and what is then the composition of HIV and how it works. I will fast forward it a little bit. Maybe this was too much. Where here we can see it's jittering sometimes a little bit unexpectedly. Here we can see by clicking what type of proteins we have in different areas. By going through this structure myself, when I was clicking inside the capsid, for example, I discovered this protein which is called reverse transcriptase, which is the protein which takes the RNA of the HIV and copies it to the human DNA. This is a protein which doesn't exist in human body because we are always forward copying from DNA to RNA and not reverse. So this the virus has to ship it with itself in order to be able to make this nasty function of copying its RNA into the human DNA and making himself so resistant then to be cured. On the other hand, this might be an opportunity for the mankind using this functionality for genetic diseases, where based on the architecture of the HIV, you can rewrite certain parts of the DNA, of the human DNA, which is susceptible to certain genetic disease. So it can be used for good purposes too. Here we see the reverse transcriptase. Okay. So, but we can, with this, not only render a single HIV virion and the surrounded blood plasma, but actually if we multiply this 250 times, it's still, we have interactive frame rates on the GTX 980 cards, I think, at that time. This amount of proteins and atoms corresponds to the E. coli bacteria, which is much bigger than the HIV, but we can see that if we will have such a model, we could already render it. But what you see also in this video, especially if we zoom out, that we do not see too much. We see basically a soup of colors, which is not very informative from a certain distance. It's a lot of computation, but not very much information. So this brings us to one project which we did after the speeding up of this process, to make the visualization a little bit more meaningful and to invent color schemes which are kind of scale aware, scale dependent. By that I mean, if you look here at this picture, for this protein, which is very close to us, if we would just show its color for protein one single color, it will be not very informative. We don't even know by this cutout what protein it is, and if it will be just everything blue, then we will not be too much informed about its structure. Here, however, if we look at the detail, we can see how does what type of atoms it consists of. So if you want to zoom in on a protein, we probably want to see what is its atomic composition. If we are zooming out, we are rather interested in maybe seeing where one protein starts and where another one ends. So we have to make a color scheme which is based on the distance from the object, showing different information. At the same time, color as a hue is quite a scarce resource. It doesn't have too much to offer if we use it categorically. Just please forget about this wheel here on the for your left-hand side. Just look at the video. You can see how it behaves. At the beginning, when we started to zoom in, each compartment had a distinct color, and all the elements inside looked pretty much rendered by the same color. They also were with the same color. But as we are zooming in, we are seeing more and more detail, and we are not seeing other parts. So we basically borrowed the colors which were used in the other parts of the scene for encoding information for a particular frame. Because we are dealing with a highly hierarchical structure, and for each distance we want to show another information within this hierarchy. So if we have compartments, that's one level. Then we would like to see the members of the compartments, which might be the proteins then. The proteins are often consisting of multiple chains, so we would like to see the individual chains within the protein. And then we are dealing with amino acids and then atoms afterwards. So, and what is this mysterious wheel doing here? That is basically just showing a occupancy of or how much the color resource, the color hue, is being used. And if it's available, certain segment of the color, then by force layout, we basically expand the usage of colors to that area for encoding information for the scale we are currently looking at. Okay, so that was already going beyond simple rendering. There were visualization questions to answer in order to show some meaningful images, not only fast renderings. Here, the second example which I want to show you is that somehow the pipeline of these biologists and also artists have changed. Previously, when they wanted to integrate knowledge, they typically would take all the literature which they surveyed and try to represent it on a single image. And for the last, let's say, 20 years back, this was still a good strategy because they were sort of up to date with the drawing when they finished it, compared to the start. These days, the rate at how often are discoveries in biology made would not be sufficient anymore, because if you are drawing an image for, let's say, several months, then after these several months there will be new protein discovered which should be part of the scene, and it's not, and then you will have to revise the scene again, which will be suboptimal. So this was one process, and within this process, then the biologists and illustrators were using these kind of atlases in order to convey information what is in the scene. And so this is a 2D kind of a cutout, but you understand that it is a 3D scene, and every element is numbered and it corresponds to certain protein or protein complex. Now, these biologists are having different workflow. They are integrating the information in the procedural recipe, which creates this 3D model. And once this 3D model is created, there is no guarantee that you take one plane and there will be all the proteins which you want to show. Otherwise, you will have to constrain the modeling then accordingly, which might be a severe constraint to a certain degree. So instead of that, you can have some visualization technique, occlusion management technique, which will help you to fine-tune the visibility of elements within the scene so that you can still convey the information content of the particular image. So as you have seen also in the previous videos, often clipping has to be made. So this is one thing which geometrically you probably have to do in order to reveal structures which are inside. So one cut or through a cutting plane would be useful, or such a cut with a kind of a box which is injected in the center of the data set could be another strategy for clipping, and so on. So, pardon me. So, and once we have this clipping, we will see a lot of structures we want to see, but we will still be missing some structures from the entire composition. And the question is how to bring them into the scene back. For that, we have developed this visibility equalizer technique, which for each instance is basically showing you how many of the proteins proteins are shown in relatively from the entire amount which is in the data set. And you can see how many are visible on the scene directly, how many are present in the scene but not visible, and how many have been clipped away. And as this widget is to your disposal, you can override the visibility, and you can bring some structures which have been visible and clipped away back into the scene, in order that you can create this illustration that gives you labeling of all the instances which constitute certain organism or biological structure. Here you can see again in a video where first a rough clipping plane is designed, and then you can bring in back those structures which have been previously clipped away. And notice that as we have the hierarchy of the compartments and so on, we can, along this hierarchy, simply bring not only one instance of proteins, not only one type, but the entire group, which is part of the hierarchy. Okay, so here again we are using something which we had been using for the acceleration purposes before. The fact that we have one type of protein, and of this one type, many instances, thousands of the proteins are there. So you can see that we are, by removing certain elements of the scene, we have a good selection, in fact, because we are not taking care of that one particular protein needs to be in the scene. The protein type has to be represented, so it doesn't really matter which particular element do I take away, and I have the liberty to make some constraints in order to have the most visually rich scene. This is in contrast to the previously cut-away designs which were based on, let's say, human anatomy. And there, if you want to see a liver, you have to show that one liver. It was only one instance in the scene, so you didn't have this liberty of choosing out of 10 livers because there was only one there. In case of the proteins, you have a choice of hundreds of thousands. You can which ones you want to leave in the scene and which you should take away. Okay. Now, this was all procedurally generated data based on the microscopy imaging and also the X-ray crystallography, as I told you before. You can imagine that this microscopy imaging can be directly used for rendering cells, for example, and this is what we are doing in one project. And in fact, we want to expand the range where the visualization is used from, let's say, hundreds of nanometers to maybe hundreds of micrometers, which is the size of eukaryotic cells. So almost the size which would be visible to the naked eye. Here you can see a typical volumetric data sets which are coming from microscopy imaging, and these can be then through fluorescence also tagged so that you know which organelle you are looking at. And currently we are developing some illustrative visualization techniques which are then conveying these organelles here. So you can see the yellow structures here would be the microtubuli, while the bluish ones would be mitochondria structures, and in the center there is the nucleus, which contains all the genetic information. So, and each structure is in fact represented in a different form. So while everything starts with the microscopy, there is a lot of processing and machine learning done afterwards. For example, the mitochondria are just represented through their center line and their thickness along this line, which makes it possible to use somehow some implicit representations instead of volumetric representations to mimic more the structure of these organelles. Similarly, here you can see these yellow strands of microtubuli, which have been also extracted with various machine learning techniques. Now, the scene currently is, this is work in progress, what I'm showing to you. So the scene is currently quite sparse. It should be dense. It should be big like this, and it should contain hundreds of trillions of atoms, which is clearly something which doesn't fit to the graphics GPU memory, so we will have to do something about that. But for now, what we can do could be an idea that if we know what a particular area or particular volume consists of, we can populate this information in the nanoscale there on the fly. So instead of having a pre-processed data sets, we just store the information about the recipe, what it consists of, and then basically on the fly we generate this geometry using GPUs. So this has been done one initial work where we, for example, are having three strategies for three different types of macromolecules. The one would be the phospholipid membranes, and there we are using some Wang tiling strategy in order to populate large areas of membranes with phospholipids without seeing any seams, and it seems to work quite quickly. The second strategy is that we are populating so-called soluble elements. So those ones which are just floating in water molecules, those would be in these compartments, and those ones can be simply populated in the scene, and you can then detect collision and resolve the collision for these elements until the scene is collision-free. Remember that we are having 50% of space covered by water molecules, so there should be a possibility to resolve the collision. And also the placement is somehow a little bit advanced in a way that there is a good starting point, that there is not too much intersection at the first place. The last thing to mention, and currently the most complicated one, is how do we populate the linear strands? And here we have only not really a parallel version for developing it. It simply takes a random walk with certain parameters. Oops. It takes a random walk with certain parameters, and so that it doesn't intersect itself. So it is first created, and then the soluble elements are placed in the areas which are still free and not occupied by these linear strands. Okay, and you can see here that the performance, the performance of what we can do, we can create in real time a lot of instances of the same virus. So just changing the random seed will create us a different scene, and it's quite fast. We have here three data sets which we tested it on. So one was just a blood serum, the plasma of 2 cubic microns. So here we will fit inside, I think, 16 E. coli bacteria, densely packed. That is the amount of atoms we have created. The second population was of this HIV. So instead of using the usual framework by the biologists, we accelerated their modeling performance again 1,000-fold. And the last example was of modeling the Mycoplasma bacteria, which has, however, a very big DNA structure. So the linear random walk takes quite a long time. I think takes 100 seconds to be generated. And if you guys have good idea how to accelerate random, non-intersecting random walk, then please let me know, because we will need a solution for that. Okay, and indeed we can, this is, as I'm telling you, just a work in progress, but we can populate this nanostructure directly in the microscopy microscopy resolution data. So, for example, here we see the microtubules, which are previously just implicit representations. Now we are populating there actually the proteins. So you can see these proteins which are forming these microtubules. And what you can see also is some kind of a flickering there, which is an artifact. And this artifact is coming from the fact that we are having a lot of scales to cross. And the resolution for the depth buffer for our near and far clipping plane, if it's set by distances in one scale, when you have enough of resolution in the depth buffer, in the nanoscale, simply the resolution of the depth buffer is not good enough. So for this, we would need to basically change the resolution of the depth buffer continuously, so that for each scale which we are seeing, it's a good, there is enough of resolution to show the structures. Hopefully, this was clear somehow. How are we doing with time? Okay, so this will be quick. So the last thing which I would like to talk about is that all what I told you was basically structural biology. There was not life in the data. It was only how it looks like. It's anatomy, but not how it works. And I will be talking about one aspect, how physiological models can be integrated in this. Then later on in the visualization session, Daniel will extend this discussion. So there are different models in biology how physiology is captured. Some are known as agent-based models or individual-based models, which I'm certain you have heard about and you have even implemented, probably some of you, where you have actual agents. These agents have certain interaction rules, and based on the vicinity, these agents are then undergoing certain interaction process. This is one way how you can implement a process. It's a bottom-up modeling. And another modeling could be top-down, that you just represent quantities which are of events which are happening, not exactly each event, but how many happened at a particular instance in time. Those are called kinetic modeling approaches, which are then represented by a differential equation system, and which describes how many quantities are changing, created, how many reactions are undergoing over time. Okay, so for this all amounts of data which we have seen, the agent-based modeling to do globally is a little bit big cake even for the best supercomputer in the world. However, these deterministic approaches, this kinetic modeling, is really just takes a fraction of time to model, because even for a larger subsystem, if you want to model it, the simulations are coming out quite fast, so that you can see the development even in real time. You can even steer these simulations. So, and we used these quantitative modeling approaches. We connected them. So we took one model or any model which is quantitative, and we basically applied on the elements in the scene, but not in a bottom-up approach as the agent-based systems are doing, but in a top-down approach. So we have every instance is first just randomly walking around, which is a cheap thing to perform, and then we introduce in our scene a god, which is then deciding based on the quantitative simulation which elements will be undergoing a reaction. So we have some notion of some structural properties of the scene, which two elements are, for example, close to each other or are heading towards each other, so that we can select those pairs for reaction which kind of seem to be the most natural ones to choose. So instead of having an agent-based system, we have a kinetic-based system, which is then applied through the God's decision on the passive agents. So the chemical reaction is happening, new reactants are then created, produced, and so on. And this is one big advantage for communicating biology, because we usually see only a fraction of the entire scene, and we would like to see something interesting in the scene. And if you are running agent-based modeling, you can never guarantee that the intended reaction is just happening in front of your camera. You will have to move the camera somewhere where this reaction is happening, and possibly if you have a post process, so you first do the simulation and then place the camera, this could be possible, but our intent is to have interactive steering, so that we can add some chemical elements into the scene, and then the organism will kind of act based on this. The models will act on it, and then the entire organism. So here we can, based on this top-down approach, we can choose that those elements which are in the camera frustrum have a higher probability to undergo a reaction, while still maintaining the global amounts of reactants which should undergo a reaction in consistency. Okay, so this is a metabolic pathways model of a human physiology. We are not that far yet, but I can tell you that actually it was, I think, 4 years ago or 3 years ago, where on a supercomputer they were able to model the entire lifetime of Mycoplasma bacteria in real time. So the entire simulation, quantitative simulation, took 9 hours of the entire lifetime, while the lifetime of the bacteria is 9 hours as well. So we are not too far from maybe getting these quantitative models for simple life forms really to be at interactive rates. We just need from NVIDIA better cards. So this is a very simple metabolic pathway here, which I want to show you, which is shown here. And this is a small demonstration how this is then implemented, how you can see the structural interaction of these reactants. What you learn here is that these big guys are the proteins, which are enzymes, which are catalyzing certain reactions, making it possible, but the reactants are in fact these small molecules often. So what you see in the metabolic pathways, these elements are typically the small chemical compounds, while at these junctions there is some protein sitting which is controlling and catalyzing this process. So this has not been done within our system, entire system for the HIV or for the Mycoplasma yet, but this is some work for the future. Okay, I don't want to give a conclusion slide. I would like to give you a slide like what could be there maybe after 5 years. So after 5 years, I hope we can scale up the models to the entire eukaryotic cell, that the entire atomic composition will be ready for visual inspection by anyone on this planet, and the visualization will be streamed online, and it will be interactive. The entire life cycle of at least simple life forms like this Mycoplasma could be done in real time, and you can see how its life evolves in silico, in fact, and you can see it in the structure, not only in the graphs where the concentration changes are plotted. What could be very interesting, and it relates to the talk of Daniel later, to create a warehouse of parts of mechanisms on the nano level, which we can maybe put together in a unique way and create new structures out of that, which can then form new machines. So, but it is up to us to create something like this. I think the visualization and graphics community must be part of it. not only the biologists themselves, they will not manage on their own. Then it would be really interesting to perform in silico experiments instead of in vivo for these life forms. At some point we will reach the level where these in silico organisms will be really, we will have trust in these models so that we can perform experiments not in vivo but in silico, and by that accelerate the biology process even more, because now setting up all this cleansing of proteins and separating it and everything like this, one experiment takes years. Now in silico you can do it within a fraction of minutes to get some hypothesis confirmed. Last topic which I have here, what could be maybe after 5 years at least we can get started there, to use such a representation as I was showing you as a visual debugger for biological computation. This topic is still out there. People want to use bacteria instead of processors to compute. And I'm certain that for this we need some new methods, how an SDK to be able to write programs for that. And we need some debugging tools as well. So our technology could be part of that, debugging framework as well. All right, so this will be all from my side for today. And this is, I'm just reporting the work. The work has been done predominantly by all these people which are listed here. I hope I didn't forget anyone. And in order to increase the coolness factor, I have added here this QR tag. So you can just find the URL of the website where the CellView system is present, which is a remote-based server, remote rendering system, so that even without the graphics card, you can explore the anatomy of the HIV at home. Thank you.